Yeah, so I think I will start. People are still entering. Welcome, everyone. For you who don't know me, I'm Rick van Nuland. I'm a program manager for Bigpicture. So some of you have received emails from me in the past. So today's webinar is on the best practices for AI use in clinical trials. Just a few practicalities before we start. So the meetings will be made available on SharePoint after this session, and the meeting will be recorded and put on SharePoint for internal use, so people can, so your colleagues who might be interested can have a look also after this session. With that, I think I hand over to today's speakers. So it's Roberto Salgado and Karsten Denkert. And I think Roberto will provide a first introduction.
Okay, excellent. Many thanks, Rick. Thanks all for attending on a Friday. It's also courageous to hear this type of lectures on a Friday near the evening. The topic is AI in clinical trials, and what we're briefly going to discuss is just why do we have clinical trials within the big picture? So why did Karsten and I did such a lot of efforts to include trial groups within the big picture? And we have a lot of good reasons to do this, and that's what we wanted to inform the community of the big picture. So in practicalities, please mute your line when some of us talk, Karsten, either I. We will hold the questions at the end of the lecture, and we will try to keep our schedule. So with that being said, Karsten, it's all yours now.
Yes, thank you very much, Roberto, Rick. It's a pleasure for me to present. I will share my screen. So you can see the first slide now. Okay, so the first topic for the seminar today would be my presentation focused on research options for pathologists in large clinical trial networks. And basically, I will give a very short 10-minute overview of the experience that we as pathologists had together with the German Breast Group for the last more than 15 years, actually. So we have made very interesting experiences, and I think it's some of this is really a big best practice for future activities. In 2011, 10 years ago, there was a commentary by George Post in Nature, where he basically said that the biomarker research must reach a new and a better level. So saying the dismal pathword of fragmented research on disease-associated biomarkers should be replaced by a big science approach. And there is a failure of researcher to do the research in a coordinated way. And he had this figure showing that there are 150,000 at that time, so much more now, claimed papers saying we have a new biomarker, and this little dot are the biomarkers that are actually in the clinic, and we do not have much more at this point 10 years later on. And there were extensive discussions how this problem could be solved, and actually what we are planning in Big Picture is also a very important step to focus on big science approaches for biomarker research. So it's very important to have the basic knowledge and to do this in a very coordinated way. So if we think about clinical trial cohorts, it's very important that the cohorts are large enough. So we would like to have cohorts with more than 1,000 samples. This is difficult sometimes, but it should be several hundreds of samples, not several dozens of samples. And in a clinical trial, it's important that adequate amount of tissue. Recommendation is 80 to 100% of patients provide biomarker tissue that could be used for translational research. And a very important step to achieve those high numbers is to introduce a central pathology so that each In this case, breast cancer biopsy is sent to a central pathology lab to validate the standard parameters such as tumor type, immune cells, and immunohistochemical markers, and then the tissue is later on stored in a biorepository. Those large sample cohorts are basically only possible for FFPE tissue. It's very difficult for frozen tissue, at least in a multi-center setting. And then we have to think about if we want a heterogeneous cohort, or if we, for example, in breast cancer, focus on a triple negative cohort. If we are planning to do this, it's important to already think about possible validations. So whenever we find something, we should have another cohort that is suitable for later clinical validations of this biomarker. And obviously, we need to have a pre-specified analysis plan as a document, and the methods we use should be analytically validated. Very general, if we think about a new biomarker, there are three issues that are relevant for us as pathologists and for the clinical researchers. So we have to think about clinical validity, analytical validity, and clinical utility. Clinical validity is a very easy first step. Anything we try to establish as a biomarker needs to have some kind of significant result for prognosis or for prediction of response in some subtypes. This is typically the very first step of research, and these are most of the biomarker papers that are published that show some clinical validity. But we need to have analytical validity, so we need to make sure, is the test really working? Can we repeat this in a different setting? Is it dependent on the pathologist as an observer? And what are the strategies for standardization? And the most difficult thing is the clinical utility, so the conclusions that we have out of this for clinical practice and research. So we want to have something that is really practice changing, where we use a new diagnostic test and it improves the treatment of the patient. So if we think about possible AI-related tools for diagnostic pathology, we need to make sure that all these three important issues are solved. And these are some papers. I will go show some data, some information from this first paper, but the other ones are also very important to make sure that the biomarker research is standardized. Together with the GBG, I received a few slides from Bärbel Felder from the GBG. We have a long history of translational research and biobanking. There was a decision in 2004 to create a central biobank of the German breast group that was at the Charité. It was then, when I moved to Marburg, the tumor bank, this was like two trucks of samples, was relocated to Marburg as well. And now we have more than 100,000 samples for translational research. From 2011 on, we have a central pathology assessment where we get the sample before the patient is recruited to the clinical trial, which is very helpful for standardized baseline parameters, but also to really make sure that the tissue is available in the tissue bank. So this is all research performed together with the GBG, the German breast group. So I have one slide explaining what the GBG is, because it's important to know this when some of you are planning to do translational research with the GBG slides and resources and digital slides that will become part of the Bigpicture repository. So the GBG was founded based on a group of clinicians from Germany with a strong focus at the University of Frankfurt, and they decided to have the GBG Forschungs Research GmbH as a legal and operating platform. So they are performing large multi-center clinical trials, which a scientific strategy board and several subboards. So the GBG has more than 600 participating sites and 1,000 investigators, and all the research ideas and the research input come from an academic setting. So it's not a company that is providing service to pharma. The ideas come out of the academic community. GBG is organizing the clinical trial activities, and of course there is funding and intensive interaction with pharma. So the financial sponsor will typically be a pharmaceutical partner, but the sponsor for the clinical trial will be the German breast group, which is interacting with many national and international study groups with sites worldwide and recruiting patients from clinical trials in Germany, Europe, and worldwide. And it's very important to open this resource to translational research, and now also in Bigpicture to research related to digital slides, to digital pathology, and to machine learning algorithms. These are some details, so it's a long way from the development of a clinical trial to the actual results. So it's something that is going on for many years, starting with the first idea, with the execution of the trial, with the analysis, and here we have the follow-up activities and translational activities and the patient outcome that is collected. And the main activities within Bigpicture will be in this area, so building on existing clinical trial cohorts and using them for translational research. And there are certain rules which make sure that this is really an independent research effort. So all the main developments, the trial management, the database, the ownership of data is with GBG, which is very open to collaboration with academic partners or research groups. This is the current status. So we have FFPE samples from probably much more than 50,000 samples centralized in our Institute of Pathology at the University of Marburg in the GBG Biobank. So these are patients. There's typically more than one patient, sample per patient, so more than 100,000 samples, and there are H&E and IHC slides, so that's more than 250,000 slides. They are all from clinical trials with a standardized treatment and available follow-up data, so they are a very important resource for translational research in Bigpicture. We have scanned actually most of those slides already, and we are doing some analysis on quantitative pathology. Here, for example, it's a project where we look on the size and distribution of lymph node metastasis in lymph nodes that are collected after the neoadjuvant therapy, where we would also expect to see some response to the therapy within the tumor tissue in the lymph node. And we are collecting approximately 2 to 5% of all breast cancer cases in Germany in the GBG Biobank. This is a very important information because I'm always explaining this to my colleagues at the other pathology institute. We are not monopolizing research. This is just a very small section of the resources. So there's enough breast cancer tissue left in the Institute of Pathology to do their own research, but for the clinical trials, it's much more valuable if this is centralized in a central biobank and can be used by many groups for their translational research projects. So we have a very efficient workflow. These are the core biopsies from the neoadjuvant situation. We have some slides, then we put a part of the core biopsy in a tissue microarray. We have some samples for RNA and DNA, and of course, those slides, the large slides, but also the TMAs are important resource for digital pathology. So within the Bigpicture network, we are somehow bridging between clinical trial research and basic research. So the translational research includes samples from the clinical trials, but they are used for exploratory investigations as they are typically performed in basic research projects. So everything that is green are elements that are important for this combination. So in a good translational research project, it's important to have a defined statistical analysis plan, although it's not done in a prospective setting as a clinical trial, but in a retrospective setting with available tissue. We are able to generate new hypotheses. Obviously, we can also validate predefined hypotheses, and we have to be somewhat more flexible with the statistics and bioinformatics if we are doing this in a basic research setting. What is also very important for an efficient network activity is that in a clinical trial setting, the authorship is defined by obviously the ideas that contribute to the development of the clinical trial, but also by the recruitment of the patients. So centers who are recruiting more patients in a clinical trial have a lot of work to do in this setting, and they obviously can contribute authors. In basic research, authorship is defined by ideas, very similar, but also by laboratory work. So it's very important if we have a translational research publication to combine those two concepts so that the people who have contributed to the clinical trial are still co-authors when a translational research biomarker project is done, but also the people that have done the laboratory work would also be part of the authorship. So it's actually really a bridge between the clinical trial world and the basic research world, and it's important to know the specific situations to do this in a very efficient way. So why are we doing this? Why is it important to focus on the clinical trial cohorts? And there's one very important publication by Simon, Pike and Heiss from 2009 that has really advanced the standardization in the field, and it was also a reaction on the criticism on biomarker studies that in many situations were not reproducible, or it was difficult to translate them to clinical practice. So what they defined is a level of evidence, and obviously for a practice-changing research, we would like to have a level of evidence of 1, and this is typically done in a prospective, predefined clinical trial to evaluate a tumor biomarker. And there are examples for those trials, and this is certainly the best way. But in many situations, it may not be feasible to finance a clinical trial that is designed to address a tumor biomarker. And then we have this category, which is called prospective retrospective studies, where there is a prospective design, but the samples have been collected in a previous clinical trial under standardized conditions. And the idea of this guideline paper is that if we have 2 of those studies with identical results, that could also lead to a biomarker that has a level 1 level of evidence and could be used in clinical practice. This is very different from what we are typically doing in pathology. So the classical way to look at pathology cohorts is to collect samples from the archive. We are also doing this in Marburg. It's very valuable for some research questions, but typically there is no standardized treatment. The selection is not standardized. So this is a level of evidence of 4 or 5. It's hypothesis generating, but it cannot change the way we are treating patients and we are doing diagnosis. And if we think about the future big picture strategy, it will be very important to have at some point those options to do retrospective, prospective validation, where we could simply have 2 studies within the big picture repository that could be used for validation of the biomarker. And it's very important to do this and to be aware of this opportunity. So if we really want to do this, this is the idea that we have developed together with the GBG to perform the translational research within the clinical trial samples. So it's planned to store the digital slides in the big picture repository. This will be very likely a large number of slides because it will be interesting to store not only the H&E slide, but also the slides related to the immunohistochemistry. There will be, however, only very basic clinical information stored together with the slides. There will be additional information on the treatment in the clinical trial, the response and the survival, which is not stored in the Bigpicture repository, but outside in the clinical GBG clinical trial database. So whenever a project that needs this data will be performed, it needs approval by the GBG translational support, but it also makes sure that it's really possible to do completely independent prospective validation, because this response information will be then later added to the, for example, the AI algorithm results that have been performed on the slides in the Bigpicture repository. So this approach provides a very good opportunity to have additional research options within the clinical trial worlds, and also validations for AI algorithms that will be generated as part of the Bigpicture network. So these are the collaborators of the GBG. Bigpicture is also a very important future collaborator, and I would like to thank you for listening to this presentation, and we will have questions at the end. So the next presentation, I think, will be directly done by Roberto Salgado. Yes, I will just say a few words. Roberto Salgado is a very famous international pathologist. I think he has the largest network of pathologists worldwide, which was founded in 2014 as a TIL Immuno-Oncology Working Group. He will talk about the activities of this group. And the idea was that it's very important to build a network of pathologists which is independent from the academic sites, independent of pharma, and that could put the experiences together. So there are very famous publications with more than 100 co-authors from pathologists worldwide that have contributed to the standards in immuno-oncology. And this is extremely nice for Bigpicture, because we can use this network to put the AI-related digital pathology experience really in the pathologist world. So Roberto, please start with your presentation.
Okay, many thanks, Carsten. Thanks all for attending again. So what I'm going to say is some experience that we have been working on together with Francesco from the Rose Group on AI within the TILS working group, exactly using the principles as defined by Carsten, using clinical trials, combining it with grand challenge narratives. And I'm going to talk actually about this, a framework for testing, validation, deployment of diagnostic imaging in pathology. And I will exemplify this notion by using the TILS, not because the TILS are so wonderful, but because the TILS are a good narrative for other morphological biomarkers. And I will start by just very briefly quoting this opinion piece published this week in The Lancet, bridging this chasm between AI and clinical implementation. And I will just quote a few of their statements. And actually, and these are very relevant if we want to avoid in a few years' time and we have spent all the money in Bigpicture, and actually nothing has changed in our daily practices. So the quotes are very rapidly: many advances in artificial intelligence for healthcare have been commercialized, but few of them are implemented in a healthcare setting, which is true. And they identified 3 items: transparency, suitability, and adaptability. And I will exemplify these 3 items during my lecture with very concrete example how important these are. In radiology, there are many approved, regulatory approved applications, but few of them are really supported by published studies. As Carsten mentioned, most of them have been using retrospective designs, limited in number, unpowered, etc. And mostly, and that's important, most of these algorithms will retrospective validation of facility that do not reflect real-world clinical practices. And that's important when we want to use clinical trials, because those clinical trials have been driving practice change. So why the TILs? The TILs are just immune cells. So the small round blue cells, what you see here, these are the TILs. The TILs look the same in every tumor type. So in that sense, it's a very easy biomarker. And I will exemplify this example using grand challenge narratives developed with Francesco from Jeroen's group. And why are the TILs important? Just 2 slides. This is a paper which is in press, and here you see the blue line. This is triple negative breast cancer, very young age population, younger than 40 years old. If you have a high number of TILs at 15 years follow-up, you have less than 2% risk of recurrence if you do nothing. So this is untreated. So you just follow these patients for 15 years and you have to do nothing at all. Nowadays, all these patients get the most extensive toxic chemotherapy. And on prediction for immunotherapy, here you see the blue line. This is more than 5% of TILs. So TILs predict immunotherapy response in a phase 3 trial setting. And on your left side, you see the levels of evidence as exemplified by Carson. So we have several phase 3 evidence already that the TILs predict response to immunotherapy. So it's one of the highest levels of biomarker evidence so far. But what are the potential issues that we still need to tackle before we could consider the TILs in our daily practices? And just a few examples, and these are the examples that we will try to tackle with the grand challenge narratives that we are developing with the TILs working group and Francesco's group. First of all, we have scored, we have defined, and it was actually Carsten who developed the method, and we just adapted it very briefly and made it a guideline at the end. But Carsten was the first one to develop the method to score the TILs on H&E slides. And we have nowadays, I think, 30,000 patients in breast cancer with the TILs have been scored and published, many phase 1, 2, and 3 clinical trials. And the question is, should an AI tool have exactly the same methodology as defined by the guideline by pathologists? So should the morphological guidance be reflected in the AI tool? And what is the ground truth? Is it the pathologist concordance with the AI tool, or is it outcome, or is it a combination of both? And how do you define this? Many of us think historically that the concordance with pathologists is actually what determines, and the better the concordance, the better the ground truth might be, but maybe it's not, or maybe it is, but maybe combined with some measure of outcome. And I will discuss this very briefly in a minute. On reporting, with AI tools, we can define an extreme amount of TIL variables, and I will show a few of them in a minute. And all of these need a special reporting. Imagine this is a report, an example that we have suggested to the community, how the TILs might be reported. And imagine giving to a clinician this report, and he will get completely nuts because he would simply ask, what should I do with it? Is too much detail clinically relevant? And that's something that we need to ask us, and that's why we need clinical trials. So can we define the ground truth? And how can we ensure a reliable integration of TILs? And you can replace TILs by any morphological biomarker for future AI implementations. How do we do this? By clinical trials. Carsten has already reported on this very famous article, and I started to read it in very detail and try to do the exercise, add some AI thinking in this narrative. And here you see, and I will not go into much detail, the typical levels of evidence. Most AI tools and most AI publications, even if they are published in high-rated journals like Nature or Science, most of them are retrospective and not powered at all for the biological question that is needed to investigate. And you can do this using mostly prospectively retrospective designs of clinical trials, which are available. They have been finished, they have been published, and you need to go, as exemplified by the GBG framework, you need to go through a process to get access to those slides and develop together with the trial groups the research around that biomarker. There are an extreme amount of, and exaggerate a little bit, there are many data reporting checklists, most, Prisma, Remark, and there are many of them. And the question is always, which one do we have to use if we want to report and document the analytical requirements, analytical purposes of our AI tool and our clinical questions? Let me show you a few examples about Remark. Remark mentions if you want to publish your biomarker, you need to define the following for the assay. You need to define a detailed protocol. How did you control for quality control, your reproducibility, your quantitation methods, etc.? When you read clinical articles about using AI tools, this is rarely mentioned in that level of detail. But if you want to use those tools in a regulatory approved setting, we have to do this. And going further about what are the statistical requirements, how do you develop your AI model so that it has a reproducible notion of how your patient could or could not be treated? And that's extremely crucial. And this type of evidence is something that we are mostly able to document using the designs of already published clinical trials. And this is for the simple reason that any AI assay is an assay like any other assay. And the regulatory authorities will consider an AI tool with the same basic principle as we would ask for the regulatory approval for immunohistochemistry or for NGS. We have the typical example of analytical validity, clinical validity, and clinical utility that we need to translate towards a narrative for AI tools in pathology. Because this is what is actually will drive practice change and acceptance by the community. So why do we propose and why are we working with the working group around a kind of risk assessment strategy for biomarker assessment in clinical trials? Risk is defined very easy, the probability of harm. And the risk assessment is the process assessing this probability. And what you see here, we have defined, and this was published in a manuscript, and I forgot to add reference, apologies, in 2017 or 16 in Lancet Oncology. There are three types of risks to consider if you want to develop an optimal biomarker and an optimal biomarker assay using clinical trials. And the reason why this risk narrative was developed is because many of the biomarkers, as Pastor mentioned, do not get into clinical practice. Most of them actually do not get into clinical practice. And what are the risks to biomarker development? The risk are the difference between, for example, central and real-world testing. Poor assay performance, how do you define this? And the concept of this risk assessment strategy is that you can define risk mitigation strategies for AI tool. And here you see an example of a risk assessment strategy that we are still developing in a manuscript that I will mention at the end of my presentation. Here you see the item, case selection and assignment procedure. The risk is that the sample size is not adequate. And if you don't have an adequate sample size, you will not be able to answer the endpoint of your study. And the sample size should be determined by the number of patients included in your study, not by the number of tiles and not by the number of slides, as an example. An additional risk mitigation strategy is that to make sure that the validation set, and Carsten alluded a little bit to it, needs to cover the spectrum of the cases that you want to have your biomarker used in the situation it is intended for, namely daily practice. Here you see different types of items and risks defined on the scanning, on the standards for imaging. Let's have a very quick look on the whole slide image standards. If you don't have standard formatting, you will limit the interoperability of the tool in different settings. So we need to have standard image formats or DICOM standards for interoperability. And what you see in your upper left, the column about tripod is just another reference checklist for AI tools. And we are trying to translate that checklist into AI applications in anatomic pathology. And here you consider another manuscript that we have developed and published more than a year ago in the Lancet Oncology about assay approvals. And this was driven by the mess and the confusion around PD-L1 assays in the community. And one of the items that the pathology community in that manuscript alluded to, look at the first one. Industry should be mandated to do concordance studies with other similar assays or standardized controls before the drug is approved. So what we are doing for this moment is we are trying to translate this narrative into an AI narrative. Look, let's go back to the first item. Industry should mandate it to perform concordance studies with state-of-the-art algorithms or standardized controls before an algorithm is approved. This is still being discussed and debated within the working group. And we are very transparent, so we will make the draft public one day so that everybody can comment on it. And let's look, for example, at the last item. Regulators should require data confirming the analytical validity of the algorithm in the distributed setting in which it would be applied to. And this was driven by the experience of PDL1 assays, where you have in a central setting, in a CRO of a clinical trial, an excellent concordance. But when you go to the daily practice in the real world, this concordance drops significantly. And this has learned us the lesson that we need to find a different way on how translating the biomarkers and the assays used to define those biomarkers from a clinical trial setting to the daily practice. And that's where grand challenges come in. On the reference standards, and this is a project which I think is extremely important as a concept that I would like to propose to the big picture, that we are trying ways to integrate it into the Grand Challenge project that we are conducting. It's a project conducted by Brandon Gallas from the FDA. And the purpose of this project, of which you will see the first set of data in a month or two, probably, is to create a data set of pathologist annotations for validating AI tools. So the question about here is, how can we ensure that different AI tools have more or less the same type of performance? When you talk to pathologists about why do they want to implement AI tools, they say, well, because we want to avoid the lack of reproducibility between the pathologists in the same center, which is reasonable. But the problem for the patient would be that patient would go to a different center, which uses a different AI tool for the same biomarker. How can you ensure that the AI tool in center B will be analytically equivalent with the tool in center A? And currently, we don't have this evidence. We don't have the reference materials to do this type of evidence. And this is something that Brandon Gallas is leading in an excellent way. So the purpose, why do we focus on TILs? In this project with the FDA, for the reasons I mentioned, it's a big topic these days. It's increasingly being implemented in clinical trials. It's increasingly being implemented in daily practice. And there is a tremendous amount of efforts on validating AI tools for the TILs. But it's not about the TILs as such, because the narratives that is being developed with this project is to create these validated datasets by using crowdsourcing of pathologists, is that one day these annotations, we make them public using a mechanism developed by the FDA so that people can use those annotations to validate your tool on and then submit it to the FDA. So that the FDA has the tools available internally to have an assessment of the analytical validity of the assay. And this is being done by the following three principles. The pathologists are trained by lectures, webinars, etc. This is the job of the working group. Pathologists are crowdsourcers from the working group to collect the annotations. And then at the end, we will compare the annotations to see to what extent the annotations are variable between pathologists. And Brandon Gallas and his team are developing specific statistical methods to be able to define this variability. So that when this data one day gets out in public, people will know the variability of those crowdsourced pathologists on the TILs in that particular material. And only then the FDA will be able to assess more accurately, and the group itself, the analytical components of their assay. So currently we have analyzed, coordinated by Brandon Gallis' group, 64 breast cancer slides, 640 regions of interest with more than 7,000 stromal TIL annotations. And the first set of data will be released in the next months. And one of the reasons, one of the most important lessons, one of the most important lessons that we have learned so far, is this variability, that there is variability between pathologists to score these annotations. And this variability can be mitigated by training. So training is crucial. As said, we want to create a data set for the purpose it is intended for, namely bridging the academic community and the work done by academic community using a clinical trials, as I will explain in a minute, to the FDA for regulatory purpose, using a procedure called the Medical Device Development Tool. And that's the main aim of this project. This will be a perfect model for all other morphological biomarkers using AI tools. So the high throughput routing project, that's how it is called. It's an FDA-led collaboration working with several different academic communities, the Pathology Innovation Collaborative Group, the TILS Working Group, and many others. And it's very transparent and public, and we involve a lot of people from industry also, because this is something that we need to do together in a very transparent manner. So what grand challenges? This is the homepage of the TILS Working Group. We have opened 2 grand challenges. I'm nearly done. And we have developed together with Francesco Scampi a platform in which we can host clinical trial slides. So we have upgraded the security levels of that platform so that we can store centrally clinical trial slides. And we can use those slides to validate machine learning algorithms. And as Carsten mentioned, the trial data are stored in the trial group, in the TILS working group in this particular study. And the important narrative for groups willing to be part of this, they don't get access to the slides or they don't get access to the trial data. The principle is very simple. The slides are uploaded on that platform. Tremendous legal paperwork needed to accomplish this. The machine learning tools go on the platform. They are run on the slides, and the data is collected and analyzed by the trial groups who have the clinical data. All in a very transparent partnership. And we are testing this model with Francesco's group in Nijmegen. Importantly, the clinical data remains at the trial site because that's a proprietary thing. But the statistical analysis is done, the statistical analysis plan is developed before the study, before the analysis of the data, and not during the analysis of the data, as is done in most research projects. So this is the model. It's actually very simple. This is the private challenge. This is accessible by the participants. Here are the trial slides. You upload the algorithms, you get an output. The ground truth is, what I will explain in a minute, define what the pathologist, which may not be true, but we will see. And then we will assess not only concordance, but we will assess impact on outcome. And we have 2 challenges. A private one, which we called the Catalina challenge. And Catalina is found within collaborative TIL validation challenge. So you just find the letters in it. This is actually in which we have, using the same platform that is stored in Nijmegen, we have stored 7 adjuvant phase 3 clinical trials in triple-negative breast cancer. Of this project, where Carsten and I participated on many years ago. And this meta-analysis has shown that TILs are very prognostically scored by a pathologist using a microscope. And we want to test if you apply a machine learning tool, do we find the same thing? That's the first question. The second question is, how can we assess analytical validity? So the working group, we have done a very big concordance study, which you see here at the bottom. 3 ring studies with a lot of pathologists in a lot of cases. Concordance is excellent. These are trial slides also from the GBG. So the Catalina challenge is applying 2 machine learning tools from 2 groups on these data sets, and we will collect the analytical and clinical validity data of those 2 machine learning tools. And actually, Francesco informed me 3 days ago that we have the data already collected in all those trials, including the RIN trials, using 2 machine learning tools. And I will show the example of one tool. These are the metrics which are developed by the machine learning tool that we will apply, that we have applied on those 7 phase 3 trials and the RIN trials from Yin Yin's group at RCR in London. So we have very different metrics available using machine learning tools, spatial profiling, clustering, and immune colocalization with epithelial cells to see to what extent does this added value of immune detail add to the prognostic importance of the TILs assessed by the pathological microscope. And my expectation would be that the more detail will not drastically change the outcome, which is already an important item that we can learn from and to see how we can take this additional detail for our patients' perspectives. And we have a public challenge which is being coordinated by Francesco, which is called the Tiger Challenge, which is now public and open. And I think there are already 18 groups registered. And with the training data, and this is different from the private challenge, which is not open to the public, which might be open one day if I get the approvals of the trial groups. These are 4 different trial groups, which is a tremendous amount of medical communication, and we will see. But this is a public challenge. 1 phase 3 trial. I will not disclose which one. And here we have developed annotated training materials using pathologists from TILS working group that we have made public. So the groups can use this publicly available training materials to validate their algorithms before it is applied on the data set, on the slide set, where we will collect the outcome of those patients. So we have in this data set, in this challenge, 2 things: a publicly available training data set and a publicly available slide set in which we can collect outcome data, but the clinical data by itself will not be made public. In conclusion, why do we need to do all of this within the big picture? Coming from the same article which I mentioned at the beginning, published in The Lancet this week, clinicians will only accept, and that's true, will only accept AI tools in their daily practices if there is a robust analytical and clinical validation are performed. If that's not the case, then Nature papers do not change clinical practice. They inform the direction of a scientific pathway. But Nature science, they do not affect clinical practice. Now, many papers have been published in New England, for example, using AI tools. None so far, practically none so far. Importantly, psychologically, and that's an important thing to discuss, is there has been literature about that algorithms are after a regulatory approval. If these are closed systems, clinicians will rarely be comfortable adapting the tool. But if they can adapt it in the future, they will be more willing to include it in their daily practices. So the more that is done, and that's the conclusion of this article, and I'm also almost nearly done with my presentation, the more that is done to address all these barriers, the more and the sooner we will see implementation of these tools in routine patient care, using clinical trials that have affected daily practice, using platforms like the Grand Challenge, and combining all of this with regulatory efforts like brand listening at the FDA. And only this combined narrative, I believe, will lead to a quantum jump in AI implementation in daily practice. And importantly, almost all what I have said is part of a manuscript that we are developing. The title has already changed. It's now called Best Practices, which started which was actually pretty good in shape 2 years ago, and then COVID came, which disrupted the lives of many of us. And this is a formally invited to Bigpicture to be part of this development. This is a paper, a manuscript that Francesco and I are developing. We will include the lessons learned from these 2 grand challenges, of which we will have the data by mid this year fully analyzed. So by the second half of this year, we would like to finalize this data, this manuscript, and I'm very willing to present the results of these 2 grand challenges, at least the private one, and Francesco maybe the public one, to the Bigpicture once we have all the data analyzed. Just a few final slides of thank you. Francesco, Brandon, James, Victor, Stefan, Balaj, Carsten, Laius, a tremendous amount of people, more than 700 people actively participating in the TILS working group. And the TILS working group is the most informal and I believe one of the most expert working groups in anatomic pathology on immune biomarkers worldwide. And we are ridiculously transparent. So if you want to know what we are doing exactly, just send me an email. A special thanks to the Tiger team who over the past 2 years in extremely difficult circumstances, for the reasons we all know, have developed the platform that we will also use for the private. Francesco, special thanks to him. These are the pathologists from the TILS working group that have contributed to all these annotated slides which I have mentioned. Thank you very much. And for any questions, please don't hesitate. Just send me an email. Thank you very much.
Yeah, thank you very much, Roberto. So we are open for discussion now. So I think the best way would be that you raise your hand. I can see it. Yes, I see there. And then you can ask your questions.
Okay.
Yeah, there was a question by Trevor McKee.
Yeah.
Hello, can you hear me?
Yes.
Yes.
Oh, wonderful, amazing work. Thank you so much. I was just, one of your last points was about, that pathologists might be more open to adapting tools if they can be modified, and yet, you need to lock them down. So I was wondering if there's any thoughts about how you actually implement that in a way that, the modifications, let's say, because the problem with machine learning is that, you could make it better. You could also potentially make it worse, depending on which annotations you feed. So is there any thoughts about, making sure that the algorithm stays robust when you're adding new annotations and doesn't sort of lead it off into a weird configuration?
Do I, Carsten, or should you?
Yeah, you just start, Roberto.
That's now, technically speaking, I don't have an answer. Conceptually speaking, I do have an answer. One of the risk, I'm a risk guy. I always think in terms of risk. One of the reasons why we developed these risk mitigation strategies at the time is that we encountered studies in which at the beginning of the study, the protocol was like that. And during the study, things started to change. And at the end of the study, the protocol, the assay was actually not anymore similar to the assay used in the beginning of the study. So these trials were clinical trials and they failed at the end. So one of the things that we might consider, and this is again the importance of translating this academic risk to the real world setting, if we find a way with the Bigpicture to define a framework in which we can prospectively do this type of quality annotations and prospectively do this type of quality comparison, so that when the regulator gets their approval out and people start using it in daily practice, we can do prospectively collection of quality metric to see, is it indeed still persisting and robust in time? And this is actually what we already do, for example, for a simple example, HER2 immunohistochemistry. So we know, for example, the percentage of HER2 amplified cases in a typical breast cancer population. So if you use an assay to detect this amplification, you should expect every year the same type of performance, even if you adapt your algorithm in the middle. So this type of continuous performance metric is something that I do think we should implement for AI tools. And so, but how to do this technically at the AI tool level, that's something for the more brighter IT experts among you.
Yeah, we have a question by Moritz Radbruch.
And just to follow up, because I think that's pretty much the most important question that was just asked. Is for the IHC of the breast cancer that you just talked about? Did you say it's there's a standard set of ground truth slides that are available for people, or did you say in-house you evaluate your own consistency with yourself, which would also be good, but kind of not mitigate the risk of kind of slow drift in one direction, but still would be pretty good?
Well, it's we know, for example, a certain percentage of HER2 immunohistochemical stained breast cancer cases do need to be amplified using a FISH technology. And so it's this type of metrics that we know already from the literature. It is this applies to all hormone receptor positive disease. We know that if you have 1,000 patients, the proportion of hormone receptor positive disease that you have. So if you apply an AI tool to do this, you need to find exactly the same. So it's this type of publicly available metrics available. The reference materials, for example, are also, and this is something that Brandon Gallis has had a brilliant idea many years ago to start to develop this in a partnership with the TILS working group, is that if you have these reference materials available out there, only then we will be able to systematically also in the future use this type of tools to calibrate every time the performance of AI tool, because it's a reference standard which we now use, for example, for other things. We have TMAs with reference standards for immunohistochemistry, for example, but we need to have reference materials for AI tools in pathology also, as we have these for AI tools for immunohistochemistry.
Do you know of any, probably this time in AI or in any other technique, a reference set that is also developing? Because that would be the next dimension, right? For example, I'm thinking of we check out a certain organ, but in the reference set we didn't think about one particular lookalike lesion, some type of special inflammation that may look like it, and we don't have it in the reference set. So we may want to add that to the reference set in the future, and so we would have both the AI evolving and the reference set evolving. Is that a thing already?
I'm an autist, as most pathologists, so I don't like change. So I think if you use a reference set, it has to be very consistent and robust and should not be changed very easily. That's why we don't have to move too fast with those reference materials development. We have to think very carefully how to use it.
Yeah. But if I may add something, if you think about the big picture concept as a whole, I think the big picture repository, once it is advanced and finalized, will become a kind of evolving reference set, because we expect to have more than one example for any pathology phenotype that exists within those millions of slides. So we must find a way to standardize this in a way and to open it so that it can be used to do exactly this, checking new ideas, new algorithms against the very rare diagnostic situations that we sometimes see that may not be within the first AI-based tool. I think that's a very important work to do within Big Picture.
So
thanks.
Rick, it's 1 minute to 4. I think, Carsten, if you agree, I think we can from our end close the session. If there are questions, please don't hesitate to send Carsten and me an email.
Yes.
I'm very happy to answer them, to try to answer them. Our presentations will be stored internally public, let's call it like that. So don't hesitate to get in touch with us. Thank you. And please follow the advice that Carsten and I gave. We need to do this, and we need to start now. As an example, and then I shut up, getting all the paperwork done for those 7 phase 3 trials to get the slides scanned, etc., statistical things, took 2 years. So if we want to have at the end of the picture something solid, we have to start now. And the metadata working group is thinking about the infrastructure of this grand challenge. So my suggestion would be, let's try to think already how we can use this infrastructure to collect the trial slides in a very secure environment. That's it. much better.
Yeah, so thank you very much, everybody, for participating. And whenever you have questions, just contact us or contact the Central Big Picture email.
Exactly. So, Roberto and Karsten, thanks a lot for your contribution. I think it was a very interesting lecture. So either contact them directly or contact through us if you don't know their email addresses. I would like to make one additional comment, and that is that we are very open for other new topics for discussions in a webinar setting. We try to organize this every month on Friday. So this was one, but we had multiple other ones, some more technical, some more philosophical. So please, if you have suggestions, come to us. It can be you presenting or just a topic that we have to find a presenter for. And then one last thing, and you have already seen that probably in your email box. So today we discussed about some barriers, and the next webinar actually on the 18th of March by Julie Swillens from Radboud will also focus on the barriers, on actually the barriers that pathologists feel they're in place when it comes to digital pathology and computational pathology, and we'll try to make it a little bit interactive, so also get some input actually from the audience. So please feel free to join and register for the next webinar as well. And with that, I hope you all enjoy your weekend and hope to see you next time.
Thank you very much. Bye-bye.
Thank you.
Bye, Karsten.
Bye.
Take care.
Thank you. Goodbye.
Thank you.